Imade University in the Institute of Food Bioresource Technology. My presentation is on effects of different wet processing methods on chemical composition of coffee of common cultivars grown in Kenya. To my introduction, coffee is a beverage that's obtained from after processing of coffee cherries. And there are different methods of processing coffee, which includes dry method, wet method, and semi-dry methods. And the quality of coffee has been may vary with the different processing methods that are used. And one factor that may be observed as contribute maybe to variations of is the elimination of, or the mode of elimination of mucilage, which is a substance that is found in coffee cherries, and fermentation is a common method commonly used under wet processing to remove this mucilage. And under fermentation, fermentation can be carried out using natural methods or even starter culture utilization to ferment this coffee, and then washing with excess water or with enough water is done. But there are other methods that do not use fermentation, and they use mechanical means to remove this mucilage. And these methods are observed to be faster, and they conserve in the utilization of water. So the use of different methods may have affects quality through maybe changes in chemical composition, and some of the chemical components that are found in coffee beans includes the protein, the fats, sucrose, and even chlorogenic acid, among others, which contribute to the quality of coffee. The study focused mainly on the determination or determining the effects of these different methods on chemical composition of coffee beans. And this coffee processing, there are also different categories of pulping methods, including maybe use of continuous pulping equipments, manual handler, and the methods or methodology, the materials which were actually the coffee cherries were harvested for the two cultivars, and then they were obtained through selective picking methods through a completely randomized design. And the cherries were sorted and pulped using different processing methods. And this is the flow diagram on how the coffee beans or cherries were processed with two of the methods utilizing fermentation, and the mechanical methods not utilizing fermentation. And then the coffee was washed and dried to the required moisture content. And this is just a photo of some of the samples that were prepared and fermented. On the dried coffee, then they were determined for components like lipids, protein content, sugars, caffeine, chlorogenic acid, and tricholine content for the samples that were processed with the different methods. And then the data was analyzed statistically, and then separation of means by LSD. So this is some of the data that was obtained, which involve protein content, sucrose, and lipid content. And from the statistics, it was noted that there were no significant difference between the different methods on these components, which are major components in coffee that contribute to the quality. Then we know that the processes, especially with those fermentation, which utilize fermentation, had no variation with those ones with the mechanical methods, which did not use fermentation. Processes with fermentation maybe could be expected maybe to, according to some authors, that some could have increased values because of degradation, like of cell wall, to increase polysaccharides, especially when it comes to sugar components. But these were not the case. We just observed that yeah, there were no variation between those that were fermented and those that were not fermented among the coffee samples. On the fatty acid composition, there were little variation in the between the processing methods on some of the fatty acid components, like linoleic acid and palmitic acid for the SL28, but though it was not common for what happens for the Ruiru 11 samples. So slightly, so we noted that there was slight variation on the levels of these fatty acids between the processing methods. So the processes with fermentation showing slightly lower levels compared to the ones that did not have fermentation. And from literature, there is indication that the metabolic processes during wet processing of coffee may affect the level of chemical components. For example, the presence of lipase activity may cause some reactions which may affect some of the components, especially lipids. Variation between coffee varieties were noted on this composition of the coffee samples. The other components that were looked at is the trichonelline, the caffeine, the chlorogenic acid, and this is the data that was obtained, and the processing methods actually that did not also show variation on the levels of these components. For example, trichonelline being one of the components which contributes to the aroma of coffee, especially after roasting, contributing to pyridines and pyrols, among other aroma compounds. So no variations was observed due to the processing methods that were used. Even with the fermentation, then there were no indication of losses of these components. So this could be because the compound are, since they are in the endosperm, then they are less soluble and they are not affected by these processing methods. Processes such as soaking, because of use of more water, has been reported to have less effects to these components. Levels in the caffeine content of the beans was not also affected by the three processing methods used. So less, because they are considered less soluble, and they are less affected by the processing methods under wet processing that is commonly used, including fermentation of coffee, has also been noted by other researchers. So it's caffeine being one of the major components contributing to bitter flavor taste, and also improving or modifying the aroma of coffee. Chlorogenic acid is another component that we looked at, and the processes did not show significant variations. Being considered as a more soluble component, can easily be lost under leaching, but there was no indication of loss for the methods that use fermentation and with excess water. So the mechanical methods and methods with fermentation did not show variation in this component. Fermentation and excess water use does not affect the level of these compounds. Yeah, and these are some of the publication we have for this information. So on the conclusion, we say that processing methods did not show much variations on chemical components, especially the protein and sucrose and lipids that we observed or we studied in this research. So processing methods slightly affected the fatty acids components, while biochemical components were less affected by the processing methods. Okay. So that's all I had for this presentation. Thank you.